Hello everyone, my name is Salman Fatemi from Mississippi State University. The title of research I would like to talk about is the effects of injection of D3 and 25-hydroxyvitamin D3 on the immune-related gene expression of the broilers challenged with the coccidiosis. Coccidiosis is one of the major economic diseases in broiler industry, which is caused by Emeria species. Previous study has shown to reduce feed intake, increase feed conversion ratio, and impair immunity and damage small intestinal morphology due to Emeria species. And just in 2009, approximately 127 million annual economic losses were reported in the United States. The first defensive mechanism is innate immunity, which is consisted of several cells, and on the surface of those cells there are receptors that can recognize internal pathogens called toll-like receptors. Those toll-like receptors induce production of the cytokine and chemicals in order to initiate pro-inflammatory response. And one of the major module or genes that contribute to activate those toll-like receptors is MyD88. And the second defensive mechanism is adaptive immunity, consists of the B cells and T cells. B cells are in charge to produce antibody, and T cells are actually the killing cells that killing the pathogen cells. Naive T cells has to turn to effective T cells. This is mediated or regulated by several cytokines such as interleukin-2, interferon gamma, and TNF alpha. Also, after inflammation and killing the pathogens, the cells has to come back to the normal situation with suppressing immunity. This is mediated by regulatory T cells or Tregs, and this is also mediated by cytokines such as interleukin-10 and TGF-beta. After intestinal absorption of the vitamin D3, it is converted to 25-hydroxyvitamin D3 or 25-OH-DD3 in the liver, and then converted to the most active form of vitamin D3, 1,25-hydroxyvitamin D3 in the renal cells. The active form of vitamin D3 is in charge for vitamin D function, such as normal calcium homeostasis, cell proliferation and differentiation, and modulating innate and adaptive immunity. As compared to D3, 25-OH-DD3 is more stable because it has longer half-life, 2 weeks as compared to 15 hours. Also, it increases calcium and phosphorus absorption in the upper and small intestine, and it stores efficiently and more as compared to D3. That would be the reason when the dietary 25-OH-DD3 at the same level of activity fed to the broilers as compared to D3 enhances innate adaptive immunity, which increases antibody production and modulating T cells activity in the commercial and infection conditions. For example, reduced inflammatory response during coccidiosis infection were reported when bird fed 25-OH-DD3 at same level of activity as compared to D3. Injection has emerged to vaccinated chicken in order to protect and provide immunity for broilers, and also it provides embryonic exposure to injected material. It can be several nutrients and vaccines. Also, it is shown it is safe and effective. As compared to dietary supplementation, it can be more cost efficient. Previous injection of the 25-OH-DD3 as compared to daily injection resulted in reduced inflammatory response, improved small intestinal morphology, as well as increases breast milk yield. The objective of this study was to determine the effects of injection of D3 or 25-OH-DD3 alone or in combination on immune-related genes expression in broilers before and after coccidiosis challenge. Water-soluble form of vitamin D sources were used, and they were dissolved in distilled water, and then transferred to 400 milliliter commercial vaccine diluent in order to reach to 2.4 microgram per egg concentration at the time of injection. All solutions were prepared cold and fresh, and 50 microgram of each solution were injected into the corresponding egg at the time of injection. 1,000 eggs were incubated in thick master sedan hatcher, and at 18 days of incubation, the corresponding egg were injected with the Zoetis evo injector machine, and at hatch, 4 males were placed in each of 8 replicate suspended battery cages belonging to each of 5 treatment groups in each of 2 separate rooms. The rooms considered as a blocking factor. The experimental design that was randomized, completely block design, and within each room we had 5 treatments. The treatments were non-injected commercial diluent, D3 and 25 OH-DD3 at 2.4 microgram per egg, and the combination of these two at 4.8 microgram per egg. At 14 days of age, birds were challenged with the coccidiosis B52 vaccine, which consists of 4 different species of the emaria, and only injection-injected treatment were injected, which means the non-injected treatment were not challenged. At 14 and 28 days of age, which is 2 weeks post-infection, 5 grams of the meal of the jejunum were selected and isolated in the liquid nitrogen for the gene expression. The RNA were isolated using the triazole protocol. Later, the quality of the RNA were determined using the nanodrop, and the cDNA will also perform using high-capacity cDNA reverse transcription kit protocol. The primers for this study were designed using primer tree software, and the cDNA with the primers were also used for real-time PCR using BioCompare quantitative real-time PCR machine. The housekeeping used in this study that was JPDH, innate immunity candidate genes that were toll-like receptor 4 and 15, and MyD88, adaptive immunity and inflammatory response candidate genes were interleukin 2, 6, and 10, and TGF-beta 4 and interferon gamma. All data were analyzed using SAS 9.4 with the ProgGLMX with one-way ANOVA method, and all differences were considered significant at p-values less than equal 0.05. As I mentioned, before challenge we had 5 treatments, and after challenge, because we didn't have non-injected, we had 4 treatments. Before challenge, we did not see any differences among immune-related genes with the treatments. And I would like to talk about most relevant result that we observed for the post-infection. On the y-axis, we have the unit for the each graph, and in x-axis we have a treatment, and the treatment are differentiated with different colors that you can see here. Interleukin 6 at 28 days of age, which is 2 weeks post-infection, was lower for bird received 25 OH-DD3 alone as compared to diluent and D3 alone injected bird. Interleukin 10 is considered as a anti-inflammatory response at this 28 days of age was higher in bird received 25 OH-DD3 alone as compared to any other treatment. The same pattern also were observed for TGF-beta 4 at 28 days of age. The summary result of this study showed that evo injection of 25 OH-DD3 at 2.4 microgram per egg increased expression of anti-inflammatory response and reduced expression of pro-inflammatory response genes during the recovery phase of coccidiosis infection. Also, previously we reported that in a similar study, the production of nitric oxide, which is one of the inflammatory indicator, were reduced for bird received 25 OHDD3 as compared to any other treatment at 28 days of age. The current results of this study showed that injection of 25 OHDD3 alone resulted in a decreased inflammatory response during coccidiosis infection. And this is important to know that increasing the interleukin-10, which is considered an anti-inflammatory response, has been shown to be associated with the reduced inflammation and increased body weight of broiler during the coccidiosis challenge. And this greater response to doing 25 OHDD3 could be due to greater expression of 1-alpha hydroxylase, which is isolated from the several immune cells that can only convert 25 OHDD3 to active form and cause the vitamin D functionality, not only D3. And the changes in the anti-inflammatory and pro-inflammatory response genes in response to 25 OHDD3 alone could be the basis for reduced heteroxyl production in those birds. In conclusion, 2.4 microgram of 25 OHDD3 can be delivered at 18 days of incubation in order to improve immune response of broiler during the coccidiosis infection. And also, I would like to thank to Mississippi State University graduate students and the farm crew that helped me to run this study. Also, I would like to thank to funding agency, and I would be happy to take if you have any further questions. Thank you.